Okay, so this is going to wrap up chapter two pretty quickly. So we left off talking about measuring the dependent variable and kind of the different factors that we have to, keep in mind. So we left off talking about sort of the cost of measuring the DV. And as I mentioned last week, some measures are more costly. So when we're using like paper and pencil, obviously that's the cheapest way to go. But if you are using any type of equipment or machinery or device, like when we're looking at physiological effects of the body, though that equipment's going to be a lot more expensive, right? So researchers are going to need significantly more resources if they're going to do something like that than if we're just collecting data, paper and pencil data. All right. So some other considerations that have to be made in terms of our participants and specifically their expectations. So when your participants have a certain expectation about the hypothesis, if they can figure out what it is that you are looking for and what your hypothesis is, they are going to do what is necessary to confirm that hypothesis, typically, meaning they're going to answer or behave in such a way that they assume you want them to, not necessarily how they would realistically, which is a problem, right? So we don't want that to happen. Remember, whenever we're doing any type of research, we're trying to prove our hypothesis wrong, not correct. We're trying to prove it wrong. So if our participants figure out what that hypothesis is and then behave to prove that correct, it's going to throw our data. So one of the things we can do is use deception. That's going to mitigate the effect, keeping in mind we still have to adhere to our ethics and our IRB review and what is approved. So making sure that our deception is not going to cause significant harm to the individual, but we can use a little bit of deception in terms of not giving them all of the details that of what we're looking for in the research study. We should also be mindful of the demand characteristics of the study. So this is any feature of an experiment that could inform the participants of what you're looking for or what the study is. So again, we want to avoid our participants knowing what it is exactly you're looking for because they're going to change their behavior. So, for example, when we talked about the Ashline study, right, he said we're going to be looking at how you evaluate different lengths of lines, which technically speaking, that isn't 100% a lie, right? It's a lie by omission. They are asking them to evaluate different lengths of lines and compare them to each other and tell us which ones are the same, right? So they are looking at that, but that's not the main purpose of the research. The main purpose of the research is looking at how they respond with those answers depending on how other people respond in the room. So that's the demand characteristics of a study, right? It basically means the nitty-gritty details of the study. We don't want our participants to know that. Now, we can also use filler items. So this is when we will put in random survey items or items on a test or a measurement that are not related to what we're looking at. So we're trying to disguise items on a questionnaire so that, again, our participants don't pick up on what we're looking at. Let's say we are looking at a difference between extroversion and introversion personality characteristics. And if every single one of our items on a survey only related to extroversion and introversion, our participants probably pretty quickly are going to realize that we're comparing those two things. And they might change their answers depending on how they want to be perceived. So if they don't want to be perceived as an extrovert or they don't want to be perceived as an introvert, they might change their answers, not because that's how they would actually behave, but simply because they are trying to emulate a behavior that they want the researcher to see, which is a problem. So we might throw in other survey items based on other personality characteristics, agreeableness, confrontationness, or confrontational aspects, happiness. We might look at contentment. Like we might throw in a bunch of filler items. So the participants might be able to tell that we're looking at different personality characteristics, but they're not going to be able to tell which ones specifically we're looking for. Some additional controls we can do for participant expectations, of course, are administering a fake treatment. So placebo effects is something that we can see in a placebo group. So essentially, their behavior changes because they know they're getting a pill and they don't know if it's the actual treatment or not. So sometimes we see a behavior change even though they're not being given an actual treatment. So we see this a lot of time in medication and drug studies. Obviously, our control group is not being given the active drug, but they're still given a pill. The pill is just a sugar pill, basically, but they don't know that that's what it is. There is a possibility that it is a treatment, and sometimes their behavior will change. So we always use a placebo group as our control group because we're trying to control for that placebo effect. Because if we see a behavior change in the placebo group, we know there's something else other than our treatment, something other than the drug or medication that's changing their behavior, right? Because they're not receiving the active drug. It's just a sugar pill. So if their behavior is still changing, then there's some other confounding variable that's impacting their behavior. So we do always need to have that placebo group so we can assess if there's a placebo effect taking place or not. Now, as far as our ethical implications for this goes, for having a placebo group, we've talked about this before, but at the end of the study, if you find in the experimental group that your treatment or your drug is effective and it does work and it does help them, you have to then give that treatment or give that medication to your control group, to your placebo group, once the research has conducted, right? They have a right to that useful medication once it's all said and done. All right, some additional controls we can look for in terms of experimenter expectations. So we as researchers and experimenters can have expectations for what data we're going to have. Of course, these expectancy effects can introduce bias, which we don't want, right? Our bias can impact the outcome of the research study. So some things that can happen, we might unintentionally treat participants differently depending on which condition they're in. So if we know if they're in the control group versus if they're in the experimental group, we might treat them differently. When we're recording behaviors, there might be subtle differences in how we interpret and record those behaviors, how we view them. And that's going to be an issue in terms of our data and how that comes out. So there are a few different ways to minimize these expectancy effects of both the participant and the researcher, because we have participant expectations, but we also have experimenter expectations. So how do we control for these things? There's two different ways. So the first one is attempting to minimize participant expectancy. This is called a single blind experiment. It's when the participant is unaware of whether they're being administered the placebo or the actual drug. So they don't know if they're being given the treatment or no treatment at all. They're unaware. So this addresses participant expectations. Okay, it does not address experimenter expectations because we, as the experimenter in a single blind experiment, we, as the researcher, still know who's in the control group and who's in the treatment group. So it at least addresses the participant expectations. However, if we want to control for both participant expectations and researcher expectations, we need to do something called a double-blind experiment. So this is where both participant and researcher are wholly unaware of who's receiving the placebo or actual drug. The way this works and the way this is accomplished, especially when we're doing any type of drug treatment, is the laboratory or the pharmacist will mix the two different medications and then put them in two different bottles. So like one's red, one's blue, and they give them to the researcher, but they don't tell the researcher which one is the treatment and which one is the sugar pill. So they just say, okay, here's your set of pills for one of your groups, and here's your set of pills for the other group. And you don't know who's your control and who's your experiment groups, which is very useful because as the researcher, when you're going through this study, you're just collecting the data as you see it, right? And you're not introducing any bias because you don't know. There might be a behavior change, but you don't know if those people are in the treatment group or if they're in the control group. So you're just simply recording their behavior changes, but there's no bias there and you don't want there to be. Like, you as a researcher are much more aware of it because If there's a behavior change in the control group, that's not good, right? So you're certainly not going to influence that behavior at all since you don't know which group those people are in. And typically, the researcher does not find out until the participants find out. So that happens at the very end of the study, once all the data has collected and everything's done. Then the pharmacist or the lab will tell the researcher, okay, the group with the red pills, that was the experiment group. The group with the blue pills, that was the control group. And then they can go back and look at their data and see if there's any significant behavior changes. So double blind is the best. That's kind of the ideal if you are wanting to control for both experimenter and participant expectancy effects. Okay, final planning considerations. So research proposals are often written prior to a study being conducted. So we do this because this is what you're submitting to the IRB for approval. You're also going to run a pilot study. So a pilot study is where we do a trial run with a very small number of participants. So very few participants, and essentially we're doing a manipulation check. So we're going to expose everyone in the pilot study to the treatment, to the independent variable manipulation. There isn't going to be any control participants. Why? Because we're trying to make sure that our manipulation actually works. We don't need control participants in a pilot study because we're not going to keep this data anyways. We're not comparing it to anything. We're just making sure that our protocol actually works as we expect it to work, okay? So this is very important. We do this with pretty much every study we do because the worst thing you can do is start a study with all of your participants only halfway through to find out you did something incorrect and you have to start all over again and you have to throw all of your participant data out. We don't want that to happen. So to sort of mitigate any mistakes that could be made, we run the pilot study and we expose all of those participants, however many we have. We expose them all to the treatment. We're doing a manipulation check. We're making sure that our independent variable manipulation works. Then we also do a more extensive debriefing session than we normally would if they were actual participants. So because this data will be thrown out, we're not going to use this in our final write-up of the data. We can talk to our participants during the sessions. We can talk to them at length afterwards. We can ask them any and every question we want to ask them, and they can converse with us however long they want, with any amount of details we want, because essentially we're trying to figure out what worked and what didn't, and we're trying to fine tune everything before we run it with our final participants. And then, of course, we analyze and interpret the data. So we're going to do a bunch of statistical analyses once we've collected all of the data and examine and interpret if what pattern of results we see, if there's a difference in groups, if the treatment is successful, and so on. This also helps the researcher decide if there really is a relationship between the independent variable and dependent variable, because ultimately that's what we're looking for. Specifically, we're looking for statistically significant results. So not just a difference in groups, because oftentimes we will see a difference in groups at face value, but then we need to run a statistical analysis to determine if it's a meaningful difference. Does it have impact, right? Is it a statistically significant difference? Just to give you a very simple example of how this works. Let's say I'm working with a family who has a kiddo who has extensive temper tantrums. And let's say they're having temper tantrums for 1 to 2 hours, 3 to 4 times a day, which is awful, right? That's taking up most of the day for the parent who stays at home with them. So we go in, we run a treatment protocol, and we get that kiddo down to one temper tantrum a day. Now it still lasts 2 hours, but at least it's only happening once a day. And at face value, you might look at that and say, well, he was having 3 or 4, which is like, 6 to 8 hours a day. He's temper tantrum, like he's freaking out. And now he's only temper tantruming for 2 hours. So that looks like a significant difference, and we've decreased the behavior. However, the parent, and I'm using like the parent as the statistical analysis, right? The parent is saying, yeah, that's nice that there's few of them, but like I still cannot have a child who is temper tantruming, screaming, throwing things, hitting for 2 hours every single day. That is not a significant, meaningful difference for me. We need to get it less. And essentially, that's what our statistical analysis computer program tells us. Same thing as that parent is telling us, like, yes, at face value, it looks like you've decreased this behavior significantly. But I'm telling you that this is not a meaningful difference. And so the computer program does that with our data, right? That at face value, this might look like there's a difference in groups, but I'm telling you there's no statistical, or there is a statistically significant difference. That's what we're looking for, that meaningful difference between the groups. And then, of course, we communicate our research to others. So we do want to, portray our data to other researchers, especially if they're in the same line of research as us. We often, before we even publish a paper, we will often go to professional association conferences where we present our findings to other researchers and the public in that realm of study. So, the APA, the American Psychological Association, has annual conferences. There's all kinds of behavioral conferences that go on. We have one here at Sac State where the students actually present their findings from their labs and their classrooms. And that's really fun to go to. It's every fall. We do that. But essentially, they're able to say, like, here's the research we're doing, here's what we're finding out so far, and here's what we sort of think that means. And then, of course, the final step is submitting papers to journals. Now, we've talked about this at length before, but these are subject to peer review. So you're going to have a panel of reviewers that essentially determine if the experimental design is strong. So you've got tight experimental control, and there's a strong demonstration or a strong relationship between the independent variable and dependent variable. But also, one of the more important things they're looking at is, does this paper actually contribute to what we already have or what we already know about this line of research? There, as many as 90% of papers submitted are rejected. And that's not always because it's not a strong paper or it's not a strong experimental control or not even because it's not a strong demonstration. A lot of times those rejections come from, well, all of this is great. The study is amazing. You guys collected great data, but we already have, 50 articles saying the exact same thing that you're saying. What is it that you're doing that is any different from everything we already know? Like, how does this contribute to the overall line of research and move the science forward? So, very few papers are actually published in the long run because that's what they're asking. Not only are they looking at the logistics of whether or not you conducted a strong experiment, but they're also asking, does this actually move the science forward? Do these results tell us something new that we didn't know before? And that wraps up chapter 9.